Well, it's Monday, which for me means it's dusting day. It's kind of one of those cycles that just keeps happening again and again and again, like the cell cycle. The other reason I bring up dusting is, do you know that most of the dust around our houses is actually our own cells? Specifically, it's our skin cells. As they grow and wear out, they just break off and float around the house and land on surfaces, and then we dust them on up. I know, maybe a little bit gross, but it's our body's natural way of replacing parts as they get worn out, especially like the skin, hair, that's how it grows, even the lining of your digestive system, blood cells. All these things are made by the cell cycle. A cell produces two cells, each of those cells produce two more cells, and so on and so on, and we just keep making cells. Plants do the same thing. There's nice little vigorously growing plant this spring. See all that nice fresh green growth? In fact, for plants, if they're not growing, they're probably dying. Their parts get worn out too, so they have to keep producing new ones, new leaves every spring. It's just part of life, but an important part that we'll get into in this episode on the cell cycle. Now, the essential concept of the cell cycle is a simple one. You have one cell and you want two, and you want each of these two cells to look exactly the same as that other one. No, this isn't some sort of weird ink blot test. It's to show that genetically speaking, and we emphasize a lot about genetics in talking about the cell cycle and mitosis. They had two of these longer chromosomes and two shorter ones, and each of the resulting cells has exactly the same setup. It's important that the genetics are passed on just right. That's the instructions for making a cell and operating it. So it's important that those get passed on right, along with all the rest of the stuff inside the cell. But the genetics are particularly important. So that's where we concentrate on. And that actually started way back in the 1800s. Here's a couple of bits of brief history about the discovery of the nucleus and mitosis. Robert Brown was the first to give the name nucleus to these dots staying in the middle of cells back in 1831. Also notable is he was looking at pollen under a microscope as well and noticed this jittery motions that moved around. And here's a simulation of it here. It's probably been distracting you for a bit. And we call this Brownian motion after him. Just this idea that all molecules are in motion while they're in fluid. This becomes really important later on as we talk about things like diffusion. Well, it was another 50 years or so until somebody noticed all these different looking nuclei to give it the name mitosis, which actually means kind of tangled threads. And so that was Walter Fleming, a German botanist. And in 1882, he published this really amazingly accurate drawings of chromosomes in motion undergoing mitosis, dividing as part of the cell cycle. One important concept all through genetics is going to be this idea of pairs. Chromosomes come in pairs. So here we have a long pair and a short pair, and that's repeated here. I want to drive this point home using pairs of shoes as an illustration. Well, our chromosomes come in pairs. We have 46 of them, or 23 pairs of them. You can see them over here in this picture of chromosomes we call a karyotype. So let's pretend by analogy that we have pairs of shoes instead. Here we have just 3 pairs or 6 total shoes. So if we were to do mitosis with these and divide them up, we would start by duplicating every single one of these shoes. And then we'd have to move them around. So we've got to get rid of that surrounding nuclear envelope. We create some microtubules and pull them apart after we line them up, circle them back up, and divide the cells. And now we got 2 closets full of the same shoes we started with. Notice in each one we have a right and a left, a right and a left, and a right and a left shoe. All good. So are there shoes in cells? No, not at all. Oh, okay. Well, if that last illustration was the right way, this is the wrong way. Cells copy all of their shoes or chromosomes and just dump them in a big pile. What would happen then? When they got separated, there'd be no way of really sorting themselves out. I mean, look up here. We have 2 left shoes. Here we have 3 shoes instead of 2. Here we also have 2 left shoes. And over here, we're missing 1. You know, if you were buying these shoes, you'd feel gypped. This would be terrible. In a cell, this would be catastrophic. Before we get into the process of the cell cycle, kind of step by step, this is one key thing to understand: sister chromatids. So when a cell goes to copy itself, it first needs to make a lot of preparations, and the first thing is to copy all its DNA. In the next chapter, we'll go over how that happens, kind of on a molecular detail, but for now we're just going to say you have 1 chromosome, and it needs to be copied into 2. But one of the key things about this is they're attached, that they don't separate, and that last illustration of the shoes explained why. So they have to stick together in order to be split up evenly. The other thing about sister chromatids is they're absolutely identical in every way. So this white piece of chalk represents 1 chromosome. If I copy it, I better get 2 of the exact same thing. Same color, same size, and everything. That doesn't work. Wrong color, wrong size. Needs to match in every single detail so that the cell that gets this chromosome and the cell that gets this one end up having the same genetic material. While they're together, we call them chromatids, sister chromatids, but once they split up, they're independent chromosomes. All right, now let's go on and look at the process of the cell cycle. The cell cycle, sometimes it's just called mitosis, but really, in the more technical sense, the cell cycle is composed of interphase, which is the preparation for division, and the mitotic phase, which is the division itself. So with that, let's look at the different parts of interphase preparing the cell to divide. We're going to assume we're starting with a brand new cell. This thing has just come out of the cell cycle and along with its twin somewhere else, and we're starting with a fresh, brand new cell. We go into the first gap phase, or G1. This one looks like a bit of a gap. It doesn't seem like much is happening, but really all these raw materials, proteins, glucose, fat, and the nucleic acids to make more DNA, all this stuff is really just coming into the cell as it gathers the raw materials it needs to grow. Next is the S phase, so that's for synthesis, and that's where we make more DNA. Now the DNA is in the nucleus, and it's really spread out. So unlike the demonstration I gave you before about sister chromatids, this is just going to make a whole bunch more DNA. There's 46 pieces of it in there for us, and so we're going to make 46 more identical pieces. There it is in the nucleus, done with the S phase. Now right at the end of S phase, the cell's going to check over all this DNA, sort of proofread all its work, make sure everything is good. If so, moves on to the second gap phase. More of the same. We're adding more of these raw materials into the cell, really preparing to get bigger now. I guess we don't need more nucleic acids, but the rest we do. We might make a couple new mitochondria, things like that. But the cell is basically growing and really getting ready to split in two. Well, now we move on to the mitotic phase, starting with prophase. A lot happens here, really getting the cell ready to split up the DNA, and this is where our focus is going to be. So you'll notice that organelles have disappeared. I've not redrawn them up here, but most of those are membranes, except for the mitochondria, which hang around. But all those membranes basically split up into tiny little vesicles. And so they're still there, they're just broken up into little pieces, and those will reform once the cells reform. So we've gotten rid of those so we can see what's going on. But now we need to start moving the chromosomes themselves. And there's a couple things that need to be done there. Chromosomes don't just move, right? They don't have little oars and sort of row themselves around the cell. That would be silly. But they do need to get moved physically somehow. They have to be pushed or pulled around the cell. And that's the job of these microtubules coming out of these centrioles. So those microtubules are protein fibers, and they're starting to grow out of these organizing centers here. And this whole thing is referred to as the spindle apparatus. So those are growing, getting ready to attach to the chromosomes, and something else has to happen to this DNA. Right now it's really spread out. It needs to condense. So as these microtubules start to grow, the chromosomes condense in something that looks a little more chromosome-like. And this is just so that they can be pulled apart without getting tangled up. So before, remember, you had 6 feet of DNA in that tiny, tiny space in each one of our cells in 46 different pieces. It'd be like this mess. And if we try to divide this evenly, just by pulling it apart, we just couldn't do it in an organized fashion. So we need something organized. Cells are nice and orderly. Remember, that's one of those properties of life. Order. Yeah, gotta stay nice and organized. Something we can move around and not get tangled up. And so these microtubules are going to keep growing, attached to these chromosomes, and start pushing them all toward the middle. And remember, each of these has a sister chromatid, those identical twins attached to one another. And this here brings us to metaphase. They're all lined up. These microtubules have done their job pushing all the chromatids to the middle. Remember, they're still as sister chromatids, and that's the magic. That's how they all get lined up and split apart evenly. It's also a really important checkpoint for the cell to make sure that everything is just so, because once they start splitting, there's no going back. Things have to happen in a really particular way in the cell, right? It doesn't just happen. There are rules and procedures. The way that these sister chromatids get pulled apart is by getting pulled along these microtubules. So they better be attached to each side. See this one down here? It's only attached one way, and so both of these would move that direction. Good thing we have that checkpoint there. Make sure it was attached from both sides. Now we're at metaphase. Things have passed that checkpoint. We've looked it over. We're ready to move on. So you can see that these sister chromatids have been pulled apart. There's no putting them back together again. The cell couldn't figure out how to do that if it tried. Each of these are being pulled to either side. And so now they're individual chromosomes. They've separated and they're moving. They kind of look like a flock of geese flying that just hasn't quite figured out that V formation. That's what always comes to mind for me. Now, one interesting detail, as these are being pulled, if we looked real close right in here, this is what's happening. So this is the chromosome, and there's a whole protein apparatus here, and this microtubule here in red is actually being degraded, all these little pieces, and there's a couple of little motor protein arms, and it's the chromosome pulling itself to the other end of the cell. So each of these are being pulled away as this microtubule starts to degrade. So that's how these are moving to the other end. And that takes a fair bit of energy. Mitosis takes energy. This is not a free process. Pushing, pulling, making stuff, that's all a really energetically expensive process. Well, the cell is nearly there in telophase. And it just needs to become 2 normal cells. Until we got the chromosomes to either end of the cell, we really couldn't divide the cell. But now that we have them over here, then we can start dividing the cell as well. And that's known as cytokenesis, right? Cyto is cell, and kinesis, like kinesiology, is movement. So there's actually more proteins, different type of the cytoskeleton now, actually pinching that cell in half. In plant cells, it can't be pinched, so they actually form a new cell wall in the middle. But in animal cells, it's actually pinched in 2. So that's the cytokinesis. But what's going on here with the chromosomes? Well, now that they've been moved, they don't need to be in this form anymore. They can start being spread out again. So that's why they look a little bit different. That nuclear envelope starts being reformed because we want to protect all that DNA. It's valuable stuff. So we want to protect it, keep it in the nucleus. That's starting to happen as well. And these organelles are starting to come back. And they're going to continue to do so and reform into mature cells once this thing is split. And there we are, 2 cells, and we're back to G1 again. Each of these cells is in the first gap phase. It's ready to start growing, then it'll make more DNA, grow a little more, then go through the mitotic phase again, and you'll have 4 cells. Since this is an animal, these cells probably actually stay attached. A lot of times we show it like this, right? One cell becomes two. Well, not exactly. We had one big cell and it split in two, and these two cells are essentially identical. So they didn't separate out quite like this, not in an animal cell anyway. One other interesting point. Let's say this cell stays in G1 and it's ready for another ride on the merry-go-round. It's going to do this whole process again and make another cell. But this cell's done, so I've had enough. Actually, this is exactly where I need to be. I'm going to just stay like this for a long time, years, decades. This then actually we call a G0 phase, indicating is this gap that isn't leading to anything else. Basically, it's just fine the way it is. It's not going to divide again. And many of the cells in your body, pretty much all your brain cells, muscle cells, most of your liver cells, are in this phase right here. It's only the cells that are actively dividing, going round and round, that go through these phases. And that affects some cells more than others. The cell cycle is an intricate process, isn't it? It's a lot of details, a lot of things going on in the cell itself. And this is happening in your body thousands of times every day just to replace skin. Which reminds me, I gotta dust after this. Anyway, I wonder if you could speculate what would happen if something goes wrong? And do you think anything ever does go wrong? What if genetically this one ended up with an extra chromosome because they didn't attach just right at the microtubules, and so this one was missing a chromosome? What if this one didn't get any mitochondria? What sort of effects do you think that would have on a cell? And we'll find out later. I'm not asking you to look it up, but if you have some thoughts and could speculate, post them in the comments or ask a question. Either way, until next time, keep enjoying life.